Good morning, students. Students, am I audible?
Yes, sir.
Very good. Today we are going to discuss very interesting topic, that is herbicide tolerant Basta, one of the herbicide which is going to be inactivated by secretion of enzyme from bacteria and one of the plant, that is the Medicago sativa, right? From that we are going to insert the gene of inactivation of the name of the herbicide is called Basta, right? So first of all, I am going to talk about what is Basta and what is the role of Basta in the field of agriculture. Then what we are going to insert for inactivation of such a Basta. It is an herbicide, right? So we are going to inactivate the name of the herbicide is a Basta in the field of agriculture, right? So some plants and some organisms which are producing naturally the gene for name of the herbicide, that is a Basta, right? So what is Basta or what is its role? We are going to see, right? First of all, let me take what is Basta. Repeatedly, I am saying Basta. It is nothing but it is a herbicide. Dear student, please understand what is Basta. Basta, it is a herbicide. Okay, what is Basta? Basta is a herbicide. Dear students, am I audible?
Yes, sir.
What is Basta?
Yes, sir.
Basta is a
Herbicide.
herbicide. Very good. It is a herbicide. Okay. So what is herbicide? Herbicide is a chemical substances which is used to control or kill herb is called as a herbicide. I repeat once again. Herbicide is a chemical substances which is used to control weed or herbaceous weed, that is called as a herbicide, or in order to eradicate herbaceous weeds in the field of agriculture. So that is called as a herbicide. Hoping that you can understand what is herbicide. Like fungicide, like pesticide, right? Like insecticide. So fungicide means there is a chemical substances which is used to control. Fungus is called as a fungicide. Like that, if any substances which is used to control or to kill pest is called as a pesticide. So like that here, herbicide, one of the herbicide which is widely used in that is called the Basta. Now I'm going to show you what is Basta, right? Dear students, Basta is like a, now this is a Basta which is a commercially available, right? When we cultivate in crops, right? This is a Basta, you know very well, right? Dear student, it is a Basta and which is used to control herbaceous weed. So it is to control simply weeds. So that is called as a herbicide. Now this herbicide can be inactivated by the production of an enzyme naturally. So when we use Basta continuously in the field of agriculture, what will happen first of all? It is going to control or destroy or kill weeds in the field of agriculture. In addition to that, the crops that we cultivate, which are going to be collapsed by the entry of such a herbicide, the Basta. Actually, Basta, as I said, it is an herbicide which is used in the field of agriculture for eradication of weeds. So while using this herbicide in the field of agriculture, what will happen? First of all, it is going to pollute. Then it causes soil pollution. Then it is also going to infect or affect the growth of the plant. Additionally, it is not only used for controlling of weeds, but it is also going to affect the growth of crop yielding in the field, right? So the crops that are cultivated in the field, which is going to be affected by the usage of Basta. So when we use Basta, I mean this herbicide, there is a chemical substances. There is a gene which is naturally occurring in order to show resistance against such a Basta. That is called as a herbicide tolerant. Dear students, hoping that you can understand. So Basta is a herbicide. By using this herbicide, it not only controls or destroys or kills the weeds in the field of agriculture, but also affects the cropping which are cultivated in the field. So in order to show resistance against this such a herbicide, so we have to insert a foreign DNA from one of the natural plant that is called as simply Medicago. Actually, Medicago sativa. This Medicago sativa is one of the plant that produces naturally herbicide tolerant gene that is called as a PPT. Here, before that, what is PPT? The Basta, one of the herbicide, repeatedly I am saying, this is a Basta, right? My dear student. This Basta, which affects the crops, right? This Basta, it's a chemical herbicide that contains a chemical agent that is called as phosphinoterazine. Phosphinoterazine. This phosphinoterazine is a chemical agent. It is produced by, or there is a production of phosphinoterazine. It's a chemical substances which is found in Basta. This Basta, I mean this phosphinoterazine, it's a chemical agent. It is determined by the gene that is PPT. No, PPT is nothing but phosphino, PP. The first P, phosphine. Second, pino, and T, trizine. So this is a phosphinoterazine. It's a chemical agent which is used as a herbicide. This herbicide is determined by the presence of a gene that is called a PPT gene. What is it? PPT gene. Now, we are going to insert the foreign gene or the PPT gene artificially in plant, right? One of the plant that is a tobacco plant. Generally, this PPT is a gene which is present naturally in one of the plant that is called as Medicago sativa. My dear student, please understand, this is a Medicago sativa. This plant which produces naturally the chemical substances. What is it? What is the chemical substance? That is phosphino. This phosphinoterazine. It is, as I said, it is a chemical agent which is used as a herbicide in terms of Basta. Now, this Basta is naturally secreted by the presence of PPT gene. These genes are obtained from Medicago sativa. Here, you can expect Which of the following plant produces resistance against Paspinothricine? Which of the following plant? What is the plant? Medicago sativa. Dear student, will you mention the name of the plant that produces naturally Paspinothricine?
Medicago sativa.
Medicago sativa. Actually, it is a plant that produces Paspinothricine naturally. Basta. It's a chemical agent which is used as a herbicide. Now, this herbicide is determined by the presence of PPT. Is a gene, right? Okay. Now we are going to insert this PPT gene into one of the plant, that is a tobacco plant, right? Tobacco. In tobacco plant, we are going to insert this PPT gene. Actually, this PPT is involved in the synthesis of, ammonium assimilation process, right? Ammonium assimilation process, right? Actually, this ammonium assimilation is a process, right, which is going to be inactivated, which is going to be incubated by the insertion of PPT, right? So, dear student, it is very important, right? Very interesting area to concentrate, right? So, along with this PPT or this Paspinothricine is a gene which is inserted into tobacco plant. As a result, this tobacco plant which produces resistance against the name of the herbicide, that is a Basta. It produces resistant power against the name of the herbicide, that is a Basta, right? My dear student, you can understand. When we insert this foreign gene, the PPT gene, which is going to secrete an enzyme that is responsible for the production of Paspinothricine. This Paspinothricine is made the tobacco plant as a resistant variety or resistant plant against the Basta. So when we use the Basta to tobacco plant, it is not going to be affected by this Basta, because this tobacco is a genetically modified plant that had been made as a resistant variety against the name of the pesticide, name of the herbicide, that is a Basta. So simply the herbicide resistant power is artificially introduced into tobacco plant by the insertion of PPT. Paspinothricine is a gene which is artificially inserted into tobacco. That's why this tobacco plant is a genetically modified plant. After insertion this PPT into tobacco plant, this plant shows resistant power against this Basta. Right? Because this tobacco plant, which is simply that shows a resistance against Basta. When we use Basta as a herbicide in the field of agriculture, this growth of tobacco plant is not affected. There will be no loss when we use this Basta. But Basta can be used to eradicate weeds around the tobacco cultivation. The place where we cultivate tobacco, which is not going to be affected by the use of Basta herbicide, because these plants are genetically modified plant that had been shown resistant power against this Basta. That is the name of the herbicide, that is a Basta herbicide. That's why this plant is called simply this Basta herbicide tolerant variety. Right? Okay. So similarly, one of the same component is also isolated. What we have isolated what we have obtained from Medicago sativa as a PPT is a gene that's going to determine phosphinothyrine. It is a chemical compound that is found in Basta. The similar compound is also obtained from the bacteria that is called Streptomyces hygroscopicus. My dear student, it is very interesting. The similar compound that is part here, PAT, part is nothing but phosphinothyrine acetyl transferase. It is also another gene. It is also isolated from the bacteria, name of the bacteria, that is Streptomyces. Streptomyces hygroscopicus. Hygroscopicus. It is a bacteria that encodes an enzyme which inhibits the growth of weeds, right? So the Streptomyces hygroscopicus, it is a bacteria. This bacteria also produces the same gene what we have obtained from Medicago sativa, that is called PAD or phosphinothyrine acetyl transferase. Phosphinothyrine. And A stands for acetyl. And T stands for transferase. What is it? Actually, it is a G. It is a G that is going to be shown resistant power against the name of the Herbicide-Basta. So these are Streptomyces hygroscopicus that encodes an enzyme which is going to inactivate the name of the Herbicide-Basta simply, right? Yes, student, please understand. So this Herbicide-Basta, it is also going to be inactivated by the secretion of enzyme which is encoded by the gene that is phosphinothyrine acetyl transferase. So this enzyme is inactivates the name of the herbicide, that is a Basta. When we insert, this is a part G, right? This is a part into plant, some other plant, right? So what is the plant? I'm going to list out some plants. What are the plant? One was first of all, let me introduce potato, right? One of the plant that is a potato. And one more plant that is a sugar beet. So when we introduce this PPT or PAT into potato and the sugar beet, these two plants have been successfully developed to show resistant power against the name of the herbicide, that is a Basta. So here Basta can be inactivated by the secretion of an enzyme, right, which is obtained from Streptomyces hygroscopicus, which is capable of inactivating the name of the herbicide Basta. When we introduce this gene into potato and potato plant and sugar beet, so these plants are successfully developed as transgenic plants. These two plants have been shown resistant, herbicide tolerant resistant variety. So when we use Basta in the field of potato and the sugar beet, right, they are not going to be affected by the usage of Basta, right? Please understand. So that is the difference between Basta, herbicide, and genetically modified plant, right? So far we have discussed two species. One is the Medicago sativa and another one is the Streptomyces hygroscopicus. So this is a bacteria, that is a plant. Please remember Streptomyces hygroscopicus. It is a bacteria that also produces a similar component that we obtained from Medicago sativa. That is a plant, right? That is a naturally occurring plant, right? So these are two plants, these are two organisms are having the similar components of gene, which are responsible for the production of enzyme. They are going to inactivate the name of the herbicide, that is a Basta. So don't want to get confused, my dear student. Please remember one of the plants, that is the Medicago sativa and Streptomyces hygroscopicus. They produce genes which are responsible for a secretion or encoding of enzyme. They are going to inactivate the name of the herbicide Basta. Simply, what is Basta and what is the role of Basta? In order to inactivate Basta, what we are going to insert and where we are going to obtain, right? So that is the thing to understand. So next we pass on to Basta is over. Next we pass on to insecticide or insect tolerance. That is called the Bidcrafts. My dear students, next we pass on to Bidcrafts. There are so many crops are there, but we have only a few crops are there. What are they? The BT crops are nothing but they are insect tolerance, insect tolerant variety. What are they? They are insect tolerant. Variety, right? BT crops. That means BT crops is nothing but Bacillus thuringiensis. It is a bacteria that produces only one bacteria that produces more than 200 toxin. Only one bacteria, just imagine, just only one bacteria which is capable of producing 200 different toxins. Now these 200 different toxins are obtained from one of the bacteria, that is the Bacillus thuringiensis. It is a bacteria. That bacteria produces, dear student, I am going to show you the nature of Bacillus thuringiensis bacteria. Right, this is a bacteria, my dear students. What is the name of the bacteria? Bacillus thuringiensis. Now, these bacteria which it produces (200) toxin. Actually, these toxins are produced in the form of endotoxin. So hence it is known as a BT toxin. Actually, this BT toxin is encoded by the presence of BTG. Simply you can say, what is it called? BDGs. I'm talking about now artificially the Bacillus members are used. The cultivation of cotton. There are so many crops are there, but a few crops we are going to make as a genetically modified organism or genetically modified plant, right? Or transgenic plant. We are going to insert the gene for BT toxin which is available in the form of crystal. So simply I can say CRI or cry genes. Cry genes. The cry genes are nothing but it is a endotoxin. It is also known as a endotoxin. This endotoxin is produced by Bacillus thuringiensis. Simply it is called a Bt toxin. Simply it is called Bt toxin. Well, this Bt toxin is used in some crops like cotton. One of the plant that is a cotton. It is Bt cotton. It is also called as a Bt cotton. Bt cotton. Second one, Bt brinjal. The second variety is Bt brinjal. And third one. So that is the third one. Dara, mustard hybrid variety. 11. So these are all the varieties which have been produced by using this Bt toxin or Bt genes or Bacillus thuringiensis. So the Bt gene refers to the first letter B, which stands for Bacillus, and the T stands for thuringiensis. This is called a Bt gene. The name of the gene, cry gene. Simply you can say crystal. This toxin is produced in the bacteria in the form of crystal. This crystal form of genes are responsible for controlling or killing insects which are found naturally in cotton plant. So they are called Bt toxin. Actually, this Bacillus member which produces Bt toxin, when we use this bacteria in the field of agriculture, they secrete a large amount of Bt toxin, which is going to protect, which is going to, first of all, kill insects which are naturally found in cotton plant. So I like to mention some insects which are causing damage to Bt crops, right? So let me list out. I'm talking about Bt cotton. As I said, Bt cotton is a transgenic variety. Bt cotton is a transgenic variety in which you find a number of insects. This is an example of Bt. That means free from the insects. When we take, when we go for cotton field, we can find a number of insects which are found in the cotton. Throughout the cotton field, you find a large number of insects naturally growing, right? So this is a variety which is developed as a Bt cotton or a transgenic plant that shows no insects, because when the insects are present, this cotton variety which secretes crystal form, I mean the CRO, endotoxin. This toxin is when the insects are laying and eating a cotton plant, right? So the toxin is entered into the small intestine of insects, and they cause toxin in the small intestine of these insects, and these secretion of toxin substances which is going to kill or destroy the insects which are growing on the laying on the cotton plant and feeding or eating. So automatically the epithelial cell of some these insects are destroyed or affected by the toxic substances. Right, student? It is very easy to understand. Now, this is a cotton plant in which you can say a number of insects, right? I'd like to mention some insects, right? Moths and butterflies, beetle, right? And gadflies, etc. So these insects, when they are sitting on the cotton plant and feeding and taking and eating, what will happen is this BT toxin. BT toxin, as I said already, BT toxin is available in the form of crystal, I mean endotoxin. This endotoxin, what will happen? It enters into the mouth and the butterflies, moth and butterflies and beetle and gadflies into the small intestine. It causes toxin. This toxin, which leads to simply damage of epithelial cell, right? So in the bacteria, in the insects, this endotoxin, when it is entered into small intestine, they cause damage to epithelial cell. An epithelial cell of intestine of moth, butterflies, beetles, so automatically it leads to death of an animal, right? So in order to understand quickly, I'm going to show you the picture that I have brought to you, right? Now this is a roll of BT toxin, right? So here you find a number of insects. So first of all, this BT crystal or BT toxin is secreted by the bacteria, right? This is a BT crystal, and the spores are ingested by the larva, right? And this is a crystal which is going to be dissolved in the small intestine of these larvae. And finally, the small intestine is affected by the secretion of endotoxin. Here you got to concentrate. Endotoxin. What is the name of the toxin? Crine endotoxin, which is going to bind to very specific midgut of a receptor. I mean any larva, any insects. Then these epithelial cell, this epithelial membrane of these larvae or these insects are going to be destroyed. So as a result, toxins or inserted in the cell membranes of these organisms. So as a result, death of all the organisms. So this is called as a BT cotton. Actually, BT cotton is a transgenic plant, genetically modified plant. Because the BT toxin, the gene of BT toxin is inserted into the cotton plant. When we cultivate this cotton variety in the field of agriculture, they may secrete a toxin, that is a BT toxin. These toxins are entered when the larva, when these organisms are laying and feeding and eating. So whatever the toxins are entered into stomach and they causes epithelial damage, and finally the toxins are inserted in the cell membrane of these organisms. So as a result, death we find, right? So this is the role of a BT toxin. Actually, BT cotton is a Bacillus thuringiensis. Genes are inserted into BT cotton, right? So this is simply something about of BT cotton. One more example I am going to show you, that is the BT brinjal. The BD brinjal is nothing but it is also one of the variety BD brinjal. BD brinjal. Bacillus thuringiensis toxin is also the gene for such a toxin is inserted into brinjal, that is called as a BD brinjal. Simply the brinjal is free from the disease, right? Free from the attack of insects. Simply I can say. So when we cultivate this BD brinjal, what will happen? There will be no damage from the insects. There will be no loss by the attack of insects to the brinjal field, right? Brinjal is commercially cultivated throughout the world, right? In our state, Tamil Nadu particularly, I can say, brinjal is one of the vegetable which is widely used in almost all the families, right? Now, this brinjal can be cultivated widely by using this BD brinjal. BD brinjal is a genetically modified variety that shows resistant power against disease or insects, right? So when we cultivate this brinjal, BD brinjal, it is not going to be reduced. That means it does not reduce its yielding. So it increases yielding capacity. Next, we are going to discuss advantages of BD crafts and what are the disadvantages of BD crafts. We are going to discuss, right? So one more variety I am going to show you, just understand. Now this is a BD brinjal. Simply you can say it is a variety in which it is also insect tolerant variety, right? BD brinjal is an insect tolerant variety. Particularly, I can say these BD brinjal can be shown a resistant power against Lepidoptera, right? One of the insects. Lepidoptera. Leucinodes. One more. Leucinodes, particularly. So these insects may be affected by or controlled by BD brinjal, BD variety, BD crafts. Particularly, I can say BD crafts can be eradicated, right, by the effect of Lepidopteron and Leucinodes. They are the two main insects, right? Please understand, they are very important. And one more variety that is I'd like to mention. In this BD brinjal and BD variety, one of the bacteria which plays an important role, that is Agrobacterium tumefaciens. As I said, it is a bacteria. I'm talking about, my dear student, it is very interesting bacteria. Agrobacterium tumefaciens. This is the Agrobacterium tumefaciens, which is mediated to transfer of gene of BD toxin, right? This is the Agrobacterium tumefaciens. It is a bacteria which is used as a mediator in order to transfer the gene of BD toxin into the field, right? Like a BD cotton and a BD brinjal. Agrobacterium tumefaciens, which is used to transfer of foreign gene. right? Please understand. So when we make normal transgenic plant, normal brinjal indoor, BD brinjal, that is a transgenic plant. Transgenic plants are nothing but genetically modified plant. What is modified? Genes are modified. Some genes are inserted into brinjal, that is called BD brinjal. So this BD brinjal is made possible by using of agrobacterium because these bacteria, which is a mediator to transfer gene from one cell to another cell. I mean the gene for BD toxin. So that is also very important. And one more variety, that is DMH. Please understand, it is very interesting one. Dara, mustard, Dara, mustard, hybrid. Dara, mustard, hybrid. Actually, it is a genetically modified plant or genetically modified crops successfully developed by the Indian scientists, Indian people, right? The University of Delhi, a craft center. It is successfully developed from craft center for gene manipulation. Craft centers of gene manipulation. The University of Delhi, right? So by the team of scientists, right? A team of scientists who created such a hybrid variety, that is a Dara mustard hybrid 11. Please understand, hybrid 11, right? This is a variety successfully developed by insertion of some genes which are responsible for controlling of or inactivate the one of the herbicide, that is a Basta. So this is Dara mustard hybrid 211 variety has been successfully developed by the scientists, by the people, by the scientists of who are working in University of Delhi, which is a government sponsored department, right? In the government sponsored department, a team of scientists who successfully created this Dara mustard hybrid variety, in which what was inserted? There are 3 genes are inserted. There are 3 genes. What are the genes? The first gene is a BOR gene. Second one, Bornase. Third one, Bornase. The third one is Basta. Actually, what are they? They are the different genes which are inserted in one of the variety, that is a mustard hybrid variety. Mustard. I heard about anyone mustard, my dear students? Dear students? Am I audible?
Yes, sir.
My dear students, did you understand what I mean? There are 3 genes are inserted in mustard. Please, will you mention the name of the genes?
Bargin,
Barnase,
Barnes,
Bornase,
Barster,
Master. Very good, my dear students. Keep it up. And these 3 genes are artificially inserted in the variety of mustard. by the people, a team of scientists who are jointly working in the University of Delhi. They artificially inserted 3 genes. These 3 genes are responsible for inactivation of one of the herbicide, that is a Basta. So simply by the insertion of these 3 genes in variety of mustard hybrid, that also simply inactivates the name of the herbicide, that is a Basta. So as I said already, Basta, it is a herbicide. This Basta, it is also inactivated because these 3 genes are made. These crab, mustard crab, as a herbicide tolerant resistant power. So simply, these 3 genes are providing resistant power to hybrid variety, that is a mustard variety. Hence, this plant has been shown resistant power against the name of the herbicide, that is a Basta. So please understand, sometimes you can expect which of the following genes are responsible for inactivating of Basta in mustard variety. If it is asked, you can mention gene. Or sometimes you can expect 2 more questions, write about some important genes are responsible in mustard hybrid variety for inactivating the name of the herbicide, that is a Basta, right? If it is asked, you can just mention there are 3 enzymes: gene, Bornase, and Basta. Right? So thank you for joining, my dear students. So one more topic is pending, that is a flavors of tomato. So we'll continue next class, right? If you have any clarification doubt, please ask. So far, I'd like to just like to review once again. We have so far discussed a simple topic. What is a herbicide and what is the name of the herbicide, and what is the nature of herbicide, and what is the source of herbicide naturally, and what is the name of the genes are inserted, isolated from the organism, and which are used for inactivation of herbicide, right? So thank you for joining, my dear students. If there is any clarification, please ask. And what are the advantages and disadvantages are given very clearly. It increases yielding capacity. It also reduces insecticidal property, and it is also used as a potential reduction in insecticidal property in the field of agriculture. Talking about disadvantages, simply its cost is so high, right? And its effectiveness up to 120 days. I'm talking about BT crops, right? Advantages and the disadvantages. My dear students, what are the advantages and the disadvantages? So there will be 3 points at the right. Please understand advantages and the disadvantages. Its cost is simply it increases yielding capacity. Yielding capacity is increased. Then it also reduces insecticidal property. Then it is also used as a potential reduction, right, in the field of agriculture. Talking about the disadvantages, the first point is that its cost is so high. Its cost is so high. Then its effectiveness up to 120 days effectiveness. Then it is going to be ineffective some aphids, some insects like aphids and jassids and butterflies, some other insects, right? So these are the disadvantages. Sometimes you can expect, right, my dear students? Okay, thank you for joining. Almost our time is over. I'm going to give you a home assignment. Please take it on and try to prepare well and read thoroughly and try to write yourself in your body, right? Thank you for joining. Please take down home assignment.